Hello, everyone, and welcome on behalf of the Board on Life Sciences. We're very pleased to have you joining us for an important discussion today. The topic is correlation and causation of life sciences systems. To give you a scope of what we hope to discuss here, we would like to understand capabilities and applications of computational analyses, modeling, and design in life sciences research. But well beyond that, we want to understand how we can begin to bridge the theoretical knowledge to practical applications across all of the areas of life sciences. And finally, and very importantly, how this new knowledge can be translated to policy and decision making. So we plan on having discussions of examples of the challenges and the applications, how these concepts can actually be implemented in generating new knowledge from examples in health and in ecosystem research specifically. So we're very pleased again to have you joining us for the discussion, which will begin with presentations, and then we will have time for questions. So today, facilitating the discussion is Sudha Parikh, who's American Association of Advancement of Sciences leader, and will help us find our way through this complex topic. Sudha.
Thank you, Anne, and thanks so much to our speakers, and welcome everybody. I hope you are just as excited as I am about the conversation that we're about to have. The amazing team here at the board has pulled together a cross-disciplinary group of speakers who are, in their own ways, all coming at this the challenge of correlation and causation in life sciences systems from different angles. And so we're going to have a chance to explore. We're going to have a chance to hear what angle they're taking, and then we'll have a chance for a conversation among them that will hopefully be catalyzed by questions from you. That will explore what are the boundaries, what are the questions, what are the big picture discussions that need to be had to really enable a field that is just getting started, a field that is across all of biology and life sciences. And so what I'd love to do is we're gonna go through each of the speakers. They're gonna have 5 to 7 minutes to lay out some thoughts for you all, some introductory thoughts. We'll go through those, and we won't ask questions. We'll let them finish their talks. As you are thinking of questions, if you are on the public video broadcast, I'd encourage you to use Slido for your questions. It's an app, and I'd encourage hopefully you know how to get to it and how to use it. And then if you are on the Zoom, I'd encourage you to use the chat function in Zoom to ask questions, and we'll try and incorporate them into the discussion. Now, the first thing we'll do is sort of start at the broad, talking about how data is used to understand causality and inform decisions. We'll get to what statistical analyses have been developed and are being used to correlate information and assess causal relationships. And then we'll take it into some individual disciplinary questions that are being addressed with some of these techniques. And so what we'll do is we'll start off with our first speaker, and I'm going to turn to Ben Rotman from the University of Pittsburgh to get us started. One thing I'll note for everybody is detailed bios are available for our speakers on the website, and so please do look there for those detailed bios. Thank you, and I'll turn it over to Ben.
Thanks, Sudeep. I just want to make sure, are the slides being shown somewhere? Oh, wonderful. So I'll just let you know when I need to advance. So thanks so much for inviting me to this. It's a huge honor. So I'm an associate professor of cognitive psychology. So I study how people learn and think about causal relationships. So if you can go to the next slide, please. I'm just going to walk through some of the main questions that this field has studied and some potential future directions of this field. So one of the main questions that's actually been studied the longest at the top here is how can people accurately learn the strengths of relationships between a cause and effect? So the way that actually all of these studies are typically done is that people are either brought into the lab or they're online on a computer and they're shown a series of cases, like we're following one person over a series of days, like in the right-hand side. And you can imagine on one day, someone exercises and they drink coffee and they're feeling really tired at work. And on day 2, they meditate and they're not feeling tired. And day 3, they meditate and they drink coffee and they're feeling tired. So we give participants these sorts of vignettes and then ask them at the end of a series of days or a series of cases to make judgments about the relationships between potential causes and effects. So one of the classic findings when there's just one cause and one effect, we were asking like how strong is the relationship, is that people can do a pretty good job of detecting correlations between one cause and effect when there actually is. But also when there isn't actually any statistical relationship, people oftentimes still conclude that there is, and that's called illusory correlation or illusory causation. Another finding is that when there are two causes and one effect or more than two causes, as you all know, one of the important things to do is to be able to control for alternative causes in a situation like that, especially if the causes are correlated with one another. And to some extent, people actually have an intuition about that, this need to control for other confounded causes. But at this, and people do control for it to a certain extent. But at the same time, people don't seem to fully adjust their estimates and actually make some biases in that process as well. So a lot of these findings are kind of like, well, people can do it to a certain extent, but not all that well. If you can go to the next slide, please. So the next set of questions in this field is how can people learn causal structures from observational data? So now the question is really not just like, here's some potential causes and one or multiple potential effects, but really they have to figure out the relationships between the variables. So this field is really inspired by the classical causal structure learning theory, for example, research on Markov equivalence and those sorts of concepts. So there are certain causal structures that people can distinguish from one another. For example, colliders, so when like two causes cause one effect versus just one cause causing one effect, which kind of relates to the previous slide. But at the same time, people have difficulty distinguishing other types of causal structures. So like a fork when one cause causes two effects, or a chain when one cause causes a middle thing, which causes a third thing. And in particular with chains, one problem that people seem to pretty reliably have is that they have difficulty understanding sort of like complete mediation. They think that the first thing still causes the last thing directly, even if it just goes through a mediator. Yeah, that's one of the sort of classic problems that we see over and over again. People seem to be much better with deterministic data than noisy data, but the problem with that is that, you know, in the real world, we're never really dealing with deterministic data. And one thing that I am particularly interested in that I think is especially relevant for actually biological systems more broadly is that it seems that for the most part, people are actually approaching these sorts of problems from trying to learn a causal structure from a dynamic system perspective rather than just a static perspective. Next slide, please. So another question is whether people can design experiments or interventions to infer causal structures. So this is really comes from research on optimal experimental design and information gain theory. And so, for example, these two causal structures on the right-hand side, if X causes Y, which causes Z, versus X causes Z, which causes Y. Suppose you're trying to design an experiment to intervene on one of these three variables to figure out which is the correct causal structure. People tend to, again, people can do this to some extent, but one particular error that they make is they tend to essentially intervene on, in this case, the X variable, which isn't actually going to provide any information because that's just going to percolate down to Y and Z or to Z and Y. Whereas the right thing to do is actually intervene on either Y or Z, which will actually be able to distinguish between these two. So this is a case of what's called confirmation bias, that you're just sort of building on your just confirming the belief that you already have rather than actually discriminating between them. The next slide, please. Last general question for the field is reasoning about the causal structure. So once you already have a belief about this is how the variables are related to one another, and I even have a belief about sort of how strongly are they related to one another, can people answer questions like in the case on the left, X causes Y, which causes Z. Suppose we know the state of X and we know the state of Y. In that case, actually, when we're trying to infer Z or predict Z, the state of X doesn't matter anymore because Y is a perfect mediator. Yet people still are biased and think that X still matters. And there's a bunch of other cases like that that's technically called a violation of the Markov assumption. And another situation that people are particularly bad about is when two causes influence the same outcome. So on the right-hand side, even when there's a positive relationship between X and Z and Y and Z, if we know the state of Z, X and Y actually become negatively correlated, and people, this is called explaining away. This is something that people are particularly bad at understanding how to make inferences in that sort of case. Next slide. So there's a lot of open questions. Two that I think are particularly important is that almost all of this research has been done in psychology experiments that are very artificial. We don't really know how well people are able to do this in their everyday lives, which I think is really important to understand to what extent like people can make good decisions for ourselves. And another question is that almost all of this research has been done with sort of everyday people without particular training. And we don't know to what extent scientists and people trained with sort of different amounts of training overcome these sorts of problems or don't. And that is my presentation. Thanks so much for your attention.
Ben, thank you so much for setting the stage so clearly. Now I'm going to turn to Jose Zubizarreta to talk to us a little bit about the statistical analyses that have been developed and are being used to correlate information and causal relationships. Jose.
Thank you very much, Sudeep. Thank you very much for having me. It's a true honor to be here. Can you please... thank you, yes. Can you please hit next? Next. One less, perfect. Thank you so much. So the main idea that I'd like to emphasize today in my talk is that in doing statistical analysis for causal inference, both randomized experiments and observational studies can be designed and should be designed carefully, integrating substance matter knowledge of the problem at hand into the statistical analysis. And for doing this, we have to be very clear about the central role of randomization in establishing causation, because it's via randomization that we can build these groups that are truly comparable, as I'm going to explain in a few minutes. And that it is really important as we move to more complicated, complex systems, as I was said before in the discussion, to establish causation. So in statistics, we tend to give a lot of importance to three things, which are called identification, estimation, replication, or sensitivity analysis. Identification is under what mathematical assumptions we can identify or see these causal quantities that live out there in the world and that we want to learn from observed data. Estimation is how to make the procedures as efficient as possible, for example, and ultimately we want to see how robust are our findings to violations to those initial assumptions. Next, please. So, perfect. What is central about a randomized experiment is that randomization is our ticket for inference, in the sense that randomization allows us to isolate the effect of treatment from confounding factors. So because of randomization, we have these two groups, treated and control groups, which are the same in expectation in every way except in the receipt of treatment, such that we can safely attribute differences in the outcomes to difference in the treatment as opposed to other things. Next, please. In this slide, what I want to emphasize is that if these samples are not only representative of each other, but they're obtained from a random population, randomly from a population, a target population, then we have a reasoned basis to extend or generalize our inferences from our study samples to other populations, which was another topic that was brought in the discussion. With this perspective, I think it's possible to start thinking about methods to generalize or transport our causal inferences, for example, by reweighting these samples. Next, please. Unlike randomized experiments, in observational studies, the mechanism by which units are selected into the treatment and control groups, into treatment and by selection from the population is unknown to the investigator. So the difficulty here is in one way or another, modeling that mechanism or modeling the outcome correctly in order to unbiasedly estimate the effect of treatment. Next. And one basic method that has been developed in the statistical literature to approximate this randomized experiment is matching, where very clearly one approximates or emulates this target trial by finding those units in the treated group and the control group that look very much alike. So the intuition here with matching methods is to find the randomized experiment that is hidden inside the observational study in terms of observed covariates. But to do it in a way that then we can get statements about the influence that unmeasured covariates, confounders, would need to have on our point estimates to explain away our significant point estimates. So here we do the adjustments in such a way that then we can make statements about violations to how units were selected into treatment according to our adjustment. Next, please. Another way of making these adjustments is by weighting, where here we're differentially weighting the units in such a way that the treated and control group are similar to each other and also hopefully similar to the target population. And a question here is, as we move to other methods for adjustment, for example, regression methods, how are those adjustments approximating these features of randomized experiments? Why is that? Because, as we also discussed previously, we want this causal evidence to be evident. We want to be able to communicate our findings in a very transparent way to a variety of investigators across fields and also policymakers. So in a way, we want to open the black box of the adjustments of the models we're fitting, and we want to know to what extent they are approximating features of randomized experiments. So here with weighting, we kind of see what we're doing with the data at hand to approximate that randomized experiment. Interesting questions, I believe, are how are regression methods, and in particular, more flexible machine learning methods, approximating this experimental ideal? Next, please. Next. So I want to highlight that there's a close interplay between experimental and non-experimental or observational studies. When we do observational studies, we want to very closely approximate that ideal randomized experiment that we would have done in ideal conditions. And conversely, when we analyze randomized experiments or clinical trials, these randomized experiments or clinical trials are, in a sense, a particular case of an observational study where the random assignment mechanism is random and known. So it can be perfectly modeled. And when there are imbalance assignments, sample attrition, convenience samples, as always typically happens in clinical trials, then we have to analyze those trials as observational studies. So that interplay is very important, and I think that it's important to integrate evidence from these two kinds of studies, which is an area of active work in causal inference. Next, please. So causal inference is about learning which treatment work but more precisely for whom, the question of personalization that we had before, when and why. The last question is the one of mediation. Next, please. In the digital trained causal inference, we tend to distinguish between two settings: point exposures, namely treatments that are applied once in time, as opposed to sequential in time, which makes the analysis considerably more complicated. Next. And these two fundamental designs, the experimental and observational study designs, next, very clearly distinguishing between what are strategies or assumptions that allow us to identify or see the parameters of interest from the methods that we use to estimate from the data that handles quantities. And two examples that I gave before are matching and weighting, but we can also use combinations of these methods with machine learning methods. So next. And very important is how we can assess the robustness of our findings to hidden biases, which is an active area of investigation in statistics for causal inference. Next. And there are, of course, many perspectives when it comes to causal inference. I find that the ones of statistics and biostatistics are close. Also, it's very related to what researchers do in economics and political science. And computer science is also bringing a very fresh perspective, but along all these areas, we're seeing kind of like an exponential growth of statistical methods for causal inference. And it's very exciting to see how these methods can be leveraged in the biological sciences and develop statistical methods also from those problems and help also to learn better from that forms of biological data. Next, that's it. Thank you.
I say thank you so much for that exposition of those analyses. That's going to be great for our conversation. I'm going to turn now to Elias Berenbaum from Columbia University. And Elias is going to talk to us about the computational tools, including predictive and generative artificial intelligence that are being developed and are being used to correlate information and causal relationships. Elias.
Hello. Give me 1 second. Can you see my screen?
We do.
Can you see? Oh, okay, sounds good. I appreciate the invitation. Thanks, Kavita, you and the organizers. It's a pleasure to be here today. I'm a professor here at Computer Science Department. I do AI and machine learning, and this intersection with causal inference. And today we'll be talking a little bit more about generative AI or causal-based generative AI. This is just a few references. I think I'm not sure if I can share these slides, but I will share these slides with you. Everything that I'm talking here today is coming from these papers, including from this paper that appears in the Proceedings of the National Academy. There's a paper review, the Pro, called The Causal Inference and the Data Fusion Problem. And I also talk about the something called the pro-causal hierarchy that helps us to understand the different formalism and the different tasks that we have in causal inference. That is this paper from 22, and more recently, the paper of my students in collaboration with Yoshua Benjo in Canada, Montreal, with the relationship between causal inference and neural nets that are the best methods that we have today to do generative type of AI. I will fast here, but I think because everyone is familiar with recent breakthroughs in data science, AI, and machine learning, their current systems are able to perform extremely well in making predictions in high-dimensional settings. In particular, there's huge progress in fields of natural language process, NLP, that in GPT is the one that is the popular method and nowadays, in computer vision as well, image recognition and so on, and you have the DALL-E and reinforcement learning that is related to decision making. I pick at random here. I will not go over all piece of news, but it's all over the place about how AI may be helping people to help in and having these robotic exoskeletons, or how AI may be helping to do high-dimensional prediction for breast cancers and even replacing one of the doctors to make the diagnosis and so on. then this is kind of amazing, and we are very glad to live in these times. I think the question for us in the scientific front is like, does this mean that we are done? And done here means maybe if you have larger computers, maybe the US government or the company should be bigger clusters of GPUs, or you have bigger data sets, we'll be able to make it, meaning we'll kind of solve all the problems. And another way to see that is what may be missing. And I would say that there are many things missing yet in this setting. And then I will kind of list some of the challenges. This is just a professor in NYU in psychology and neuroscience believes that saying that COVID-19 should be a wake-up call for AI, saying how bad we did and how, in some way, I would say, with all respect, how useless we were and how things went quite badly in reality in terms of we're not helping much in this sense. There's all kinds of problems related to, we can do very well predictions in terms of diagnosis of some type of cancers, but we can explain very badly what is really going on there. When we are kind of putting our hands and making some automated decisions or trying to make automated decisions, how we can be quite unfair and amplifying the biases and the disparities in the communities. All kind of other news about why AI struggles to grasp cause and effect relations. And this is a very good piece by Brian saying what AI still can do in the MIT Technology Review. Then I would say that the challenges, but taking kind of one step back of being critical and trying to diagnose this or understand what's the problem, there are still serious foundational issues in the AI side of the equations. And the current AI systems suffer from lack of having any understanding of the system or explainability capability of why they are doing the things in the way that they are. They are quite prone to unfair and unethical decision-making. In reality, I would say could elaborate, but during the questions, but they amplifying the biases that are already present in the data. They are very data hungry and sample inefficient. They are very poorly robust and very hard to generalize to a different setting than the one that was covered by the data. Whenever you have within the setting of the data set that you have, things may be okay, but when you go outside the support, things would maybe go quite bad. And also the missing generative capabilities that are related to the GPT and settings like that. Those are very thorny and hard problems. And all things that are in common between these problems is a need or in reality the lack of causal inference capabilities. Now we like to move towards having more causal-based generative AI. I introduce here an object that I think it helps us to understand what are the kind of tasks that we have in AI. I'm sharing the reference here. This comes from a book that I recommend by Pearl and Mackenzie called The Book of Why. That is some type of survey of the field of AI and causality, I would say, from hundreds of years until today, culminating AI, and also the first paper that I mentioned there from my group. And the causal, I will call the PCH, the pro-causal hierarchy. There are different levels of queries that you like to ask for the data. The first one is association. If you're trying to do prediction, it's very related to the activity of seeing. How would seeing X change my belief in Y? Or what does the symptom tell us about the disease? We can do quite well in this one. Most of advances in AI in the DNN, deep neural nets, are related to doing better and better predictions and high dimensions. Now we have a second layer that is more complicated or more challenging that is related to the effect of interventions or doing. What if I do X? Or what if I take the aspirin? Why will my headache be cured? There is some formalism in AI machine learning that's called reinforcement learning that helps us to do that in some way, a very specific way, if I can elaborate later. But still, it is one type of it's one layer of the hierarchy. The other one is called counterfactuals. This is like X prime, maybe the Joe is a unit, Joe took the drug, and Y prime is Joe is that. Then he took the drug in the factual world and he is gone. And then we can wonder and ask counterfactually, would Joe be alive that is Y, that is the opposite of Y prime, had he not taken the drug? This is a true counterfactual. There is some type of collision between what happened in this reality versus what we would like to, this alternative counterfactual reality. Very related for the AI system to the idea of imagination, retrospection, and introspection, these Y type of questions. What if I had acted differently? I didn't, but what would have happened? There's no real counterpart of that in machine learning. There is structural causal models. That is the formalism that allows us to answer this type of, or at least model this type of questions and understand if you can answer that depending on the assumptions. Now, typical ML, just to understand where we are in ML here, they usually have this layer one is related to observing. If you have input data, the input is data that is coming from layer one, of this level one. This is most of the data, 99% of the data available today is coming from layer one. But the output of the analysis, what we're expecting, or the tension that was mentioned earlier, is like most of the time you want to make a query or answer a question about layer 2 or layer 3. Let me start with layer 2 here. That most of the inference are about causal effects, about the effect of new policies or treatments or the effect of decisions. Then the research methodological questions here is how to use the data that is collected from layer 1, that has this, the name is confounding bias, to answer questions about layer 2. Now, this is just a picture here that this is, maybe there is a reality that is out there, that is unobserved, and there is a causal model there that maybe we don't even have access to, but it exists. Now, this model has the potential of generating these different layers or these different types of interactions. One is related to the observations that, as I said, is 99% of the data that we have. Even the big data is coming from this side of the equation. But there is also the layer 2 that is related to the effect of interventions, and layer 3 that is related to these counterfactual distributions. Most of the time, you don't observe layer 2 and layer 3. Again, as I just said, most of the data is the red type. Now, what is the problem of generative AI? In reality, we would like to train, for example, a neural network with, in a big cluster here that we have in Colombia, for example, we have there in the national academies, and it does have the potential of generating all the layers, but doing training, learning, we'll spend maybe a few weeks trying to use this red type of data and learn the parameters of this model in the right side. I don't observe the left, and I learn the guy in the right. And if you are successful, after maybe a few weeks of training, this is very computational intensive, this model that is the M hat, is not the true one M star, M hat predictions for layer 1 is matching the left side that is the M star there. But now, what about what's going on with layer 2? It is still empty, and this is what I call fundamental problem, is under what conditions inference in the right side that is in the M hat are valid. When do they recover the distributions induced by the true guy in the left that is the M star? And that's the problem that we have to answer. In reality, people in the field is trying to understand the different assumptions that we should put in the neural net, for example, in the right side, in order to be able to get the guy here that is yellow, similar to the one in the left side. Now, I could go on, but I would just say that from this perspective, we have all the desiderata that we like from the causal generative AI. The new generation of AI system should provide the capability of having some level of causal understanding and the capability of articulating explanations, to have more efficient and precise decision-making, to be generalizable and robust inference, and having the causal generative capabilities in addition to doing model learning. If I have 1 more minute, I just want like to address one. Do I have
wrap
one?
it up relatively quickly, Elias. Yeah. Relatively quickly. Thank you. Sounds good.
Sounds good. I will not go slowly here, but this is just for the task of decision making. This is in the context of some kind of cancer regime, the decision making, or what we call the dynamic treatment regime. We are trying to decide which kind of treatment you get in these 2 time slots here. This is X1 and X2 time, and this is the states of the patient and Y is the outcome. And then you have this unobserved confounder that is affecting the whole system. Now, if I'm doing pure randomization, that is the guy, I just focus on the guy on the right here. This is a typical randomized trial. This is the kind of regret that we have. Regret is equal to the, if you have our oracle minus what you did, is how many mistakes we are doing. And you like to have some kind of log, something that is getting stable after you make some mistakes in the original allocation of the decisions. This is kind of the red is the typical randomization. The blue is the one that if you leverage this causal structure, you kind of start getting this type of behavior that after, I don't know, a few thousand patients that you are learning how to make the decisions for the patients, things will be stable or you converge, which means that you kind of learn how to make the decision. And the green one is the one that if you are using the observational data from layer 1 that is biased in somewhat clever way, you can speed up a lot the FDA trial or the randomized trial here if you're trying to do. I think this was raised by Nathan maybe, or related to personalized regimes. Then I think causality can help in how to make the allocation, how you leverage this confounded and biased data in order to streamline decision making or improve decision making. I think I'll stop here for now. Thanks.
Terrific. Thank you, Elias. And I think we'll want to probably dig into that model a little deeper in the question period. All right, let's turn now to Eleanor Murray, Ellie Murray from Boston University, who's going to talk to us a little bit about how causal inferences are being used in public health. Ellie.
Great, thank you, and thank you for the invitation. So I thought a lot about what to talk about today in terms of bringing the focus to public health and medicine, but you know, there are a number of implementations I could have discussed and things we've seen go wrong in those. But I thought it might be more useful to kind of bring the bigger picture of what are those challenges and barriers that have been faced when we're trying to incorporate AI and machine learning into doing causal inference. And if you'll go to the next slide. Sorry. So this kind of the key points that I want to get across is that even before we add machine learning and AI into the mix, it's really hard to estimate causal effects in public health and medicine. And this is because we are dealing with people, and that really limits the types of experiments that we can do on them. You know, there are some things we can try out with randomized trials, but whenever we're thinking about anything that could have possible harmful effects, we're not going to want to do that in terms of assigning people to those. And so we really have to rely on observational data a lot in these settings, even though it has all these potential problems. And so, you know, a lot of the effort around causal inference is how do we overcome these challenges of the data that we have to make sense of it, given the limitations around what we can actually do with it. And I also think it's always useful to bring to people's mind the idea that when we're talking about causal inference in public health and medicine, that we're literally talking about life and death decisions. And I often see, you know, people kind of coming from more of the tech side and kind of thinking, oh, it would be great, there's all this data, we can play around with it. But when we're thinking about what are we going to do with a decision that comes out of either a statistical process or a machine learning AI type process, you know, if we make the wrong decision there, potentially people are going to have their lives pretty severely affected. And so thinking about, you know, what are we actually comfortable doing there? And this actually relates to one of the things that was brought up in the initial session of just sort of putting ideas in the parking lot, where one of the challenges for this is that until we're really confident that we understand how AI or machine learning algorithms work and what they're doing, and that we've got some good guardrails on them, we're always going to be having to validate them against benchmarks generated from clinicians, because we'll be looking at past data and saying, in these situations, what would the AI have suggested? And as a result, the AI can always only do as good as people, because if it does better than people, we wouldn't actually know. We would just see a disagreement between what the doctors did and what the AI is suggesting. And until we actually are intervening to give the AI suggestions to people, we can't know if those suggestions actually would result necessarily in a worse or better outcome. So I think these life and death consequences here do have some important implications for how we're going to regulate these and how we're going to validate these AIs. And so when we're thinking about just estimating causal effects in general in public health and medicine and in all of the life sciences, really the best chance at getting a good, valid estimate of a causal effect is to be as clear and transparent as possible. And the 3 talks kind of before this were all really kind of talking about what are those details in terms of what things are we having to assume either mathematically or conceptually or in terms of our prior knowledge in order to estimate a causal effect. And so those are the things that we really want to be clear and transparent about. And the challenge of machine learning and AI is that it's not really a separate solution. It more just kind of adds a layer of black box uncertainty on the process that removes this transparency. If you could go to the next slide. Yeah. So in terms of causal effect estimation in public health and medicine by both human processes and AI machine learning processes, the thing to really think about is that we need to understand 3 big aspects. So the first is the data, and this is issues around what data are available and who, when, and where does it come from. And there are things in here about issues around, are we do we have systematic bias in who we're collecting data from or what we're doing in the world that is making its way into our data. It also here is issues like, are we going to have to deal with data shifts? If we have data that we've trained our algorithm from, but it's from 2 years ago and the situation has changed now, we can see even without AI, with other kinds of statistical modeling, that sometimes historic data is actually not a good input for our models to understand what's going on in the current situation. We also need to think about sampling and measurement error and always who is left out of the data. And then we need to understand the decisions. The decisions. And this kind of goes to what someone said also in the initial setting about what is the comparison, right? So who's making decisions? What types of decisions are we trying to inform? What kinds of information is available to people who make decisions and who's left out of the decision-making process? And then lastly, what options are we comparing in terms of understanding our questions really specifically? Are the things that we have in our data the same options that we want to assess? And where and when and to whom do our questions apply? And then the piece of how are we answering it, which is all the sort of statistics and computer science that's been talked about so far. Next slide. And so kind of several of the big issues that we've seen in terms of prior implementations of AI and machine learning in public health fall into these categories. First, that when we train our algorithms, we have to train them on existing data. And our existing data contains all the biases from a sociological definition of that word that we currently have in our society. So if there's racism or sexism or other types of discrimination happening in the world, that's going to be baked into the data. And if we're not careful, then our algorithms will learn how to do that, and they will then perpetuate those problems. The second piece is that we don't make medical and public health decisions on a purely utilitarian basis. It's not just a simple trade-off of, well, this person will have more years of life saved versus this person will have less years of life saved, for example. And there are moral and ethical aspects of our decision-making process that are always happening, but these are not captured in our data. And so the AI or machine learning model can't learn them. And we need to think about how do we build guide rails on the outputs of these processes in order to make sure that we're not ending up with some suggestion like never treat anyone over the age of 65 because they have less years of life left than someone under the age of 65, which would not be within our moral and ethical standards. And then a last part is that a lot of the data that we have, especially the data from trials and other experimental settings, specifically excludes groups that we consider vulnerable populations from a medicine and public health perspective. And this is because we want to limit the harms these groups are exposed to, and we consider that therefore research has potential harms and we should limit the research on them. But as a result, this means we have less data on these groups to allow the AI or machine learning system to learn sort of more group-specific strategies for these populations. And so one really important example for this is that we don't typically include pregnant people in randomized trials unless we're really actually just studying the effect of something in randomization. And so what we saw, when the mRNA vaccines rolled out for COVID, there was this gap in information where pregnant people hadn't been included in the trials. And now there was a lot of concern over whether this meant that it wouldn't work in pregnant people or not. And it really was just a lack of the trial evidence. Next slide. And so when we're kind of evaluating a process that includes AI and machine learning, and even ones that don't, kind of 4 questions here to sort of frame for yourself in terms of thinking about how do we actually use the answer that is coming out of this process? So whose data are we collecting and whose data is being missed? What data are we collecting and what data is being missed? How well are we quantifying the data, and what subjectivity is introduced by our quantification? And this is things like, if we're thinking about years of life lost, one of the big questions in research that uses that metric is, should you start that at counting that at birth? Should you start that at year 1? If you start that in the 3rd trimester of pregnancy, you can completely skew your answers. So thinking about these things and how we quantify them really can matter. And then lastly, what are we missing when we attempt to automate our algorithms in terms of what parts of the decisions are just not written down anywhere and are much more sort of implicit to how we're doing these processes. So I'm going to end there, and I'm really looking forward to the discussion.
Thanks, Sally. That was incredibly helpful. I'm going to turn now to Rebecca Safran from the University of Colorado. And Rebecca is going to talk to us about how causal inference is being used in ecosystems. So we've gone from public health, now we're going to ecosystems. Rebecca.
Hi, if we can get my slides up, that would be great. Can everyone hear me okay?
We can.
Awesome. Just by way of introduction, I'm an evolutionary biologist, but integrate across several different disciplines in biology, including ecology, behavioral ecology, genomics, genetics, and physiology, and come with a very deep interest in understanding sort of how to match up the specific details of individuals within populations to this broader understanding of how we get all of this biodiversity on our planet. So I just wanted to give a bit of an introduction to how we think about handling this complexity, which is from a process to pattern perspective, thinking about the mechanistic basis of how and why populations diverge in the ways they do, and how that helps us understand these larger scale patterns of species and what defines their boundaries. So if you could go to the next slide. And I think just listening to the other speakers, there's a lot of details in the data that I think we all sort of worry over collectively in terms of understanding the problems of what to measure. What you can infer from the measurements that you're taking has everything to do about the inferences you're going to make. And so the considerations that we consider is, if we're going to measure certain traits of individuals and populations, we need to understand something about how those interact with their environment, the genetic and environmental interactions, the genetic basis of that trait, before we start making inferences about its role in explaining large-scale patterns of divergence. Timescales are a really big deal. So Ellie just chatted a bit about using historic data and considering 2 years to be historic. In my field of evolutionary biology, 2 years is absolutely nothing. And so we tend to think of things on a very deep time scale, whether we're constructing these phylogenetic models to infer evolutionary history, whether we're using time-calibrated data from fossils, or whether we're making inferences on a contemporary scale. We also have the complexity that is presented by geographic variation. And so considerations for how we're going to replicate our sampling across different spatial scales is really important. And again, I sort of see this as a theme that was brought up in other aspects of the previous speakers' talks. Particularly if one is interested in how populations become adapted to different environments, one has to appreciate that the environments themselves are incredibly dynamic, and to build in some sort of replication for temporal sampling then becomes incredibly important. Layered on top of that complexity is the fact that evolution itself is a really dynamic process. How do you measure it? At what scale do you measure it? Are you measuring it just at the genomic level? Are you measuring it at just the morphological level? Are those morphological traits being tracked through genomic changes in the population? And of course, I think something that worries all of us is, getting this sort of scale up from really specific aspects or attributes of individuals and populations to more generalizable inferences, is of great concern, as well as the notion of predictability. At what temporal and spatial scales are any of these mechanisms that we're discovering actually predictable? So next slide, please. Just want to tell you a little bit about a study system that we've been working on for several decades, and some of the problems that arise when we're trying to understand these larger scale patterns here of this group of very closely related populations of birds that were actually brought to the northern hemisphere in their current distribution by humans. These are birds that build their nest exclusively on human structures. And so their evolutionary history is very closely tied to ours. And because their expansion was so rapid and the evolutionary change within several thousands of years, we can start understanding the mechanistic basis of why populations look the way they do, as well as their current distribution and its particular role on understanding the evolutionary history of this group. Next slide. And so in terms of understanding the mechanisms, we do a lot of fine scale experiments in contemporary populations through the understanding of the genetic basis of traits, the role of the environment in shaping patterns of gene expression, and or influencing how traits become adapted to their environment, and of course the role of selection and or more neutral evolutionary processes. Next slide, please. And I would say that in evolutionary biology and in the discipline that I work within, most scientists are focused on interpreting process from these larger scale population patterns of, say, population level genetic divergence and understanding which traits are associated with various signals in the genome and inferring process from there. Rather than understanding the mechanistic basis of how adaptation might be working. And it's typical then that, next slide, please, these really different scales of analysis from, on the one hand, really fine scale experimental studies within populations, considering individual variation and how that's shaped by selection, and these larger scale emergent properties are completely isolated in terms of understanding their interrelationships. But it is my view that in order to better understand the causal relationships between evolutionary dynamics and its interaction with the environment and the genetic basis of traits, that we need to start unpacking the underlying mechanisms of these larger scale patterns. Next slide. And so I just want to highlight a paper that was inspired by a postdoc in my group, Dr. Zach Laba, and in collaboration with Wei Peng, who's an epidemiologist. And this is a paper that also was co-authored by Ellie, one of the other panelists. And in midst of all the biological complexity and all the different problems and issues that we have with individual level variation and trying to understand the boundaries of different populations, whether or not they're species, whether or not we should consider them as targets of conservation, we came up with this workflow of how it is that one could step through various statistical analyses tied with various questions to better make inferences about the causal relationships between all of these interrelated elements and the question of interest. I don't want to bore you with all the details of this figure, except to say that I would consider causal inference to be very casually used in my field. And I think that this task of trying to come up with a uniform workflow is one of the first to try to at least organize us in terms of coming up with a systematic way of analyzing and interpreting causal relationships from these long-term complex data sets from population genomics, phenotype divergence, and more. So I will stop there. Thank you. Thank you so much again for the invitation. It's an honor to participate.
Thank you, Rebecca, and thank you for that, for going through that. I look forward to actually talking more about that in the questions. All right, let's turn to our last speaker, Wenying Shou from the University College London. And Wenying is going to talk to us about causal inference and how it's being used in microbial communities. So public health, ecosystems, and now microbial communities. Wenying.
Yes, can you see my screen?
We can.
Great. So I'm Wenying Shou from UCL, and we are interested in microbial ecology and evolution. We all know that correlation does not imply causality. So if we have a tube of bacteria that's exponentially growing, and we have another tube of yeast growing exponentially, if you do Pearson correlation between the two time series, the correlation coefficient will be very high. But we know that there's no causality whatsoever, no causal relationship between those two tubes, because if I did anything to the bacteria tube, nothing would happen to yeast. So this highlights high correlation does not necessarily imply causality. Now the problem is this problem is easy because we know bacteria and yeast are growing two tubes but what if they if in the same tube we again see this exponential growth of bacteria and yeast? And if we do Pearson correlation, the correlation coefficient is very high, 0.98. But how do we know that the high correlation is meaningful? So if we can do a trial to a second tube, and then we can correlate the bacteria from trial 1 with yeast from trial 2. And we see that that correlation coefficient is also high. And then we can repeat this many times, and we get a distribution. And so we can define a meaningful correlation. A within trial correlation is meaningful if it is extreme compared to cross-trial correlations. And so by meaningful, what we mean is that the population sizes of bacteria and yeast are statistically dependent. So meaningful correlation, the statistical dependence is linked to causality due to a principle called Reichenbach's common cause principle. So if they're dependent, then bacteria can cause yeast. That is to say, if I perturb bacteria, then yeast population would also change. And this can be direct or indirect. And we can have yeast cause bacteria, and we can have something causes both. And so this is why inferring causality from correlation is hard, because it is an inverse problem. That is, we are trying to deduce mechanisms from observations. And now this is highlighted as an example of why I'm interested in this is we always want to infer microbial interactions from dynamics. So for example, if microbes, so microbial communities are very important to human health and they interact in diverse ways. So if we have ground truth of these microbes, the 8 species of microbes interacting in this specific way, we then would get some dynamics, right? With some perturbations thrown in, so we get each species would be one of these curves. So now what we have is actually, in fact, those data, these observational data of microbial species. The question is that, could we get back to this ground truth? And so in this particular exercise, the problem is made easier by if I tell you that the mathematical model structure is logical Volterra, is pairwise logical Volterra model. So now, so this is what happens, right? So you have, so you would have you would have a model that recapitulates what you observe. So the matching is beautiful. But if you look at the underlying structure, there's nothing like this, right? So here, for example, 3 activates 8. So if you increase 3, 8 would be promoted, but here there's no connection. So you see that even though the data matching are good, you have same data but different models. So I highlight again the difficulty of causal inference from observational data. And because of this, controversies often arise. For example, in this paper, it says dynamical evidence for causality between X and Y. And then there's questionable dynamic evidence for causality between X and Y. And of course, the authors rebutted ferociously by reply this, like this robustness of this causal effects. So as a person who's not a specialist in causal inference, these kind of back and forth arguments are often confusing because each side seems to be, reasonable, seems eloquent. And then we were, so little story here. So we were actually given this, the task. So our colleagues wanted to understand from time series data, the microbial time series data, the interactions. And they said that, basically they said that they have tried to use some method for 2 years and didn't get anywhere. So we tried to, so that's when we actually started digging, or particularly Alex Yuan, a student in my lab, started digging into the field of causal inference. And we find that this field has very diverse, origins. It comes from philosophy, computer science, economics, and physics. So many different fields also are interested in this problem, and for understandable reasons. But actually, to understand that, it's highly non-trivial. So it took Alex maybe 1 or 2 years, and he's fast, right, to actually understand from different perspectives what they're thinking about causal inference. So we wrote this synthesis article called Data-Driven Causal Analysis of Observational Biological Time Series, and it's published in eLife. So while writing this, we actually realized that the challenges, right, the challenges of this. So challenge 1 is the traps of overselling, right? So this is especially rampant in hyper-profile journals. And so what happens is that assumptions and pitfalls are often hidden in supplementary data. And for benchmarking, friendly scenarios are used. So, and I'm not trying to say people intentionally committing fraud. It's sort of like if you had some scenarios and you see your benchmarking, your method is working, like there's no incentive for you to actually try more to figure out the failure mode, really, right? So the possible solutions is I would suggest for methodological papers, they should state conditions for validity. In fact, separately, explicitly is a box in the main text. And then the authors should demonstrate pathology when validity conditions are violated so that the readers would be better informed. And then, make statements such as, these are known failure modes. And because we don't have a proof, additional failure modes, may exist. Statements like that. So challenge 2 is like math is often inaccessible. So when we were reading this causal inference literature, we came across a method that is based on a famous theorem called Tarkin theorem. So I just want to take a second. I want you to take a second looking at this theorem, right? It's like 6 lines, no big deal. But if you look at it, it's just laden with jargons. Say compact, manifold, diffeomorphism, generic, embedding, right? So like 6 lines have like five really technical terms that are not familiar to people like quantitative biologists like us, right? So actually this theorem originates from topology. And so for people like us who have some math background but not in topology, this is a formidable task, right? And so some work is based on this. So then you're not sure how to judge, how to link the theorem to the causal analysis method. So for this, possible solution is to encourage, right, cross-disciplinary communication efforts. So for these can include head-to-head comparisons among different methods, and there are many of them, or conceptual articles that use examples to illustrate failure modes, or focus on data science tasks for which there are standard methods, but people in your field don't know. or solid solutions that have appeared in the last, say, 5 years. Existing methods, focus on these existing methods that generally have sketchy theoretical foundations, and the things like all your colleagues want to solve, but have been proven to be impossible. So we don't want to waste time trying to invent perpetual motion machine. So even statements like this would be important. And then I would suggest, of course, we need to reward these communication efforts by acknowledging the significant expertise and efforts required. It is highly non-trivial to do that. And then reward with fellowships and academic positions. So thank you very much for your attention. Oh, sorry. And then this last one is thanks to Alex Yuan's work. And thanks for your attention.
Thank you, Ying, and thank you for giving credit where it's due. All right. I want to first of all, I just want to thank everybody. You all lived up to more than expectations. That was a really interesting and provocative and illuminating set of discussions. So now I'm looking forward to the conversation that we're going to have afterward. And one of the things I'll say my big takeaway is this is hard. And I think you all said that at least once. And so that's good. We want it's an important problem and it's hard, and we're going to do this because it is hard. So now we're going to open it up to. I'm going to remind everybody who is on, who is watching us online. Oh yes.
So they're hard and inevitable. Then it's what can we do? Let's tackle it, right?
Yes. So I just want to remind folks who are watching online that if you have questions, to please use the Slido app. If you're watching on Zoom, please use the Zoom chat function. And then if you're here in the room, please raise your name tag so I can call on you. I want to open up the floor. So if there are questions, please go ahead and raise your placards. And I'm also willing to ask a first question if that's worthwhile. All right. If when I get the ball rolling, don't be shy. So the first thing I have to ask is, for all of you, the input data is extremely important. You've all mentioned it. And I noticed just in my own sort of observation of your talks that in some cases that input data is coming from the world. And so it's very hard to control. In some of your cases, though, it could be controlled by the experimenter. And so I'm curious, what levers do you all have as the folks who are trying to make the distinction of causality to get better data? Are there levers? What should the scientific community be doing to ensure that folks understand what is quote good data, and is it the same in every field? I just don't know. I'll open up that. Please raise your hand if you have something to say about that.
I'd like to jump in here because I think there's one particular type of data that the lack of it in the literature I've really been struggling with in some recent projects. And this is high quality null findings. Because when we think about putting the assumptions into our causal analysis, it's actually more important to understand what things are not causally related, because things that are causally related, we can then figure out more from the data. And there are so few really well done null studies out there. Also studies where sort of the answer is really obvious, and everyone who is in like practice, especially like in medicine, like if everyone in medicine knows something is true, it tends to not be in the literature. An example of this recently for me was I'm working on a trial that looks at knee osteoarthritis. And there's absolutely no literature on the impact of home accessibility and number of stairs to your bedroom on the decision to get a total knee replacement and the long-term pain, even though that will affect your pain and it will affect your decision to get a total knee replacement. And the clinicians are all very aware of this, but it's not something that's recorded in data, and it's not something that's being studied because it's really obvious. So obvious things and really good quality null results. I think those are the data that, at least for me, are really needed.
Thanks, Ellie. That's really important for us to hear as board members. Others? Jose and then Rebecca.
Yes, I think that another aspect is to bring methodological experts from different disciplines at the time of the data collection effort. So, for example, a lot of improvements can be done in randomized experiments by more efficiently assigning units into treatment and control. So there can be huge payoffs beyond complete randomization by doing some form of optimization and randomization together in random assignment. That translates into fewer dollars spent for the experiment, less patients exposed to a potentially toxic treatment, and so on. So I think that if we could integrate those kind of like disciplines at the time of data collection, we're going to get much better data.
Thank you, Jose. Another really informative point. Rebecca, turn to you.
As someone who's tied to sampling animals that are wild in the complex real world and all of the issues related to that, you know, we're often just faced with constraints as to what it is we can collect. And so one of the things that we always consider before we go out to collect any particular data set is what the downstream analyses are going to be, planned way ahead of our first data point. So I think that speaks to what Jose and Ellie were talking about a bit, of just having these, very standardized workflows in a sense to try to minimize all the noise of the real world, at least, has sort of helped us try to feel like we're not just, you know, picking apart pieces of very noisy data sets.
Great. Thank you, Rebecca. I'm going to turn to Nathan here in the room. And if as folks ask questions, please just introduce yourself to the whole Zoom.
Yeah, this is Nathan Price, Chief Science Officer at Thorne Health Tech, BLS member. So great session. Really learned a lot. If time permits, I'll come back in and ask some more questions. One thing that I'll start with is from Jose's talk, this notion I thought was really interesting of, within observational studies, there's a randomized experiment hidden inside, and these two quotes you gave that were super interesting about, kind of going back and forth there. And you talked about, how you could try to pull out the randomized trial by matching and, controlling for certain factors. There's always an art, I think, about, which factors you want to control for in terms of making them similar, but not throwing away signal that you might want, and kind of what falls on which side of the line. I just love maybe a little bit more in-depth conversation, open it up to the panel about, kind of the pitfalls, challenges, opportunities with, kind of looking for that proxy of a randomized trial in observational data, things to think about. And we'd just love to have a little bit more discussion around that.
I'll let the panel just chime in. And if you're on the panel and I didn't call on you, please just speak up. The hybrid way, I don't always see hands.
Thank you very much for that question. First of all, I wish to emphasize that there's no silver bullet. There's no substitute for a randomized experiment. Causation is ideally established by experimentation, to be precise, randomized experimentation. But in the context of observational studies, we want to, I think, very clearly approximate that ideal experiment. And although across disciplines we tend to agree in approximating that target trial, I think that when it comes to the statistical methods we implement with the data at hand, some methods have more traction towards that ideal experiment than others, emphasizing things like, for example, that the adjustments are an interpolation of the data at hand as opposed to an extrapolation based on a model that can be wrong, and things like that. So that, for example, also that the structure of the adjustments are very kind of like open, overt. It's easy to talk about what the methods are doing. With some methods, it's easier to do that than with others. And now your question was about how should we think, I think, about confounding. And here, I think that there are kind of two perspectives, and I think it's important to integrate them. One perspective says that we want to integrate substance matter knowledge of the problem at hand into the statistical adjustments. So namely, we want to sit down with the doctors, we want to sit down with the biologists, statisticians, computer scientists, epidemiologists, and do the adjustments kind of like prioritizing those confounders that we believe are more important than others. We want to talk about the outcome processes, the selection processes, and in that way, adjust, match, weight in a way that we prioritize certain covariates as opposed to others. The other perspective tries to learn these models directly from the data in a purely data-driven way. I find that it's very exciting. I think that the two approaches are not excluded. I came from the former tradition. I'm more keen to using methods that more naturally integrate that substance matter knowledge as opposed to kind of like black box learning the models from the data. Yeah, but I have to say that it's exciting how these methods from machine learning can learn aspects of the unknown fundamental models for the assignment process and the treatment. Sorry, and the outcome, both models, the treatment and the outcome. So those are some thoughts.
Can I say, ask or add a little follow-up to that? Really insightful comment, Jose. I think a lot of this has to do also with the construction of models that are based on biological intuition versus being driven by statistical significance. And I see a tension of that in the literature just as an editor or a researcher trying to publish empirical findings. What do you do with, after you're sitting down, say, with a doctor and, knowing that this would be a really important mediator variable to put in your model, but it doesn't have much statistical explanatory power? Like, how do you deal with that, tension or complexity in building your models in really different ways?
Yes. First of all, let me acknowledge that there are other experts in this panel who can say very interesting things about this question. So, for example, Elias knows a ton about these things. So let me offer a bit of a perspective, and maybe then he can complement what I'm saying. So first of all, a confounder is a variable that affects both the treatment and the outcome. For those variables, we want to adjust in some way. So that's an important observation. Another important observation based on your question is to try to differentiate between the timing of these variables. So variables that are measured before the treatment, we call them covariates in general. A mediator is something that is in the pathway, using the jargon from directed acyclic graphs between the treatment and the outcome. So it's after the treatment. And those variables are more complex. In general, I would say we don't want to adjust for them, in general, okay? But coming back to the covariates, when we think that a variable is a confounder because it affects both the treatment and the outcome, then there are different forms of adjustment we can do on those variables. For some variables, we can exact match on the other extreme, we can just mean balance, and that depends on the outcome model that we are in a sense conjecturing for the data at hand. So then the conversation with the doctor, say, would be about covariate prioritization. So those are some thoughts, but this is a very central question. So I'd like to offer others to comment on this.
Right. Yeah. Maybe Elias, and then maybe Ben might have some thoughts on that as well, I suspect.
Yeah, I can speak a little bit. Thanks. The first one thing that I would say is, like, I wouldn't frame as randomized control trial is the kind of benchmark and what we want. In reality, we are trying to learn about reality, and randomized control trial is one specific way of probing reality. And there are implications of this observation, but I think it's obvious what we're trying to do is learn about nature, about reality, and there are different methods of doing that, different types of measurements, different perturbations, and so on. But in other words, I think reality or nature should be the baseline. But even in the context of randomized controlled trials, there are problems with those. I think it was already alluded to earlier. But these usually the results I spent years working on the topic that's called transportability, but usually these results will not generalize to other populations. Then there is this fetish about the trial, but the trial is working in a very, very specific population, and which is almost never the population that you want. But anyhow, and then there is a whole question which is not very different than the fundamental question about which kind of assumption should you kind of supplement or should you augment the trials in order to generalize to other populations that you may be more interested to. Another consideration, I would say, consider, this is not far-fetched or not Elias is saying, but you consider the case in physics that the models are pure, the most, I would say that is the most successful use case of from the scientific perspective about how you probe reality comes from physics. And most of the data that is coming from physics, at least traditionally, maybe if you get 400 years ago until 100 years or 50 years ago, are kind of observational data. Then you stay 300 years playing with observational data, and you have the more successful use case for science, in my opinion. Then it's then how did they do it? Were they much better, or how is it possible? In general, they didn't have the fetish with the randomized trials, because it's totally impossible to do a trial regarding how planets are moving related to each other or other kind of physical phenomena. And they are thinking seriously about the causal models and the mechanisms, which kind of people are not trying to want to avoid that. And there is this thing about, anyhow, avoiding thinking hard about it. But now if the issue is about thinking hard, the questions about what is the causal language and how do you do causal modeling? And I think the fundamental question, I would say, and related to the first one about how do you get better data, I would say 99% of the data, maybe 98 will be observational anyways. Then let's just grow up, say, this is the data that we have. You want to make the claims about layer 2, about the effect of interventions or policies, or layer 3 that is counterfactuals. But now how do you do causal modeling in a more serious way and think hard about these issues? And let's understand about what is the causal language and what is causal modeling. I think this would be my general comment about the two previous, I think, questions that were raised, starting from Sudeep, if I understood his question about the data. Yeah, be happy to elaborate.
Thank you. Maybe Ben, and then Wenying, I have a question for you as well. Ben, go ahead.
Sure, although I see Ellie keeps on raising her hand, so maybe you should jump in because it seems like you have something that you really want to say.
Ellie, go ahead.
I guess, yeah, kind of going back to Nathan's original question, I think one of the nice things about that slide that Jose had with the quotes is that the second part, that within every randomized trial, there's an observational study hiding as well. And I think that really highlights in my mind, the value of this sort of target trial framework to thinking about study design is that there's some ultimate sort of theoretical ideal way in which we would answer the question that we want to answer. And that might look like an ideal randomized trial, or it might look like something else. And any real world randomized trial we're doing is still in the real world. And even a lab experiment is still being done in the real world. And if we don't think about what's the temperature in the room that the lab experiment was done at, we might miss real world implications that are happening there. And so we can have these biases creep into randomized trials and experiments that are the same biases that creep into observational studies. It's just that certain types of biases are less common in randomized trials. Observational studies are going to be prone to a little more biases, more prone to some biases that they all share. But if we think about that, we can also see that at the sort of end of that pipeline, we can also think about not just emulating a target trial with our observational study, but emulating a target trial with our actual trial, or even emulating a target trial with our complex system simulation model to say, if we were to simulate what kind of data we would have seen in a perfect trial that we would have liked to do to answer this question, what does our knowledge tell us that data should have looked like? And that this is a way that we can kind of think about those like interactions between different layers of information, and also more sort of complex causal questions like how do things impact a whole population and not just what is the direct effect on an individual.
Perfect. Thank you, Ellie. Ben, actually, if you hold on just 1 second, let me just see if I can get to another question, and then we'll let you go at it first, if that's all right. All right, Mauro.
Thank you. That was a great session. So it seems that for the examples that we hear, randomized, double-blind examples, there are two possible outcomes. But I was wondering, in other biological problems where we have to deal with the innate complexity of biology, how far we are where essentially we have multiple possible causes, not one, multiple possible causes that could lead to the same effect. Let me give you an example. So you have a deletion in p53, and eventually you have cancer. If you do a proteomic or RNA-seq or protein-protein interaction, there might be possibly in the order of 100 or potential causes that could lead to the effect. So I was wondering in the panel, essentially, whether we have or how far we are to be able to have computational tools or methods that would allow us to determine when the answer is no, yes or no, but when the answer is yes, for 3 or 4 possible outcomes.
Great. Maybe if I could turn to Ben first on this one, and then look to the other panelists.
Actually, I'll pass the buck to someone who's more computational than I am for this.
All right. Who was that one? All right, Rebecca.
Yeah, in the field that I'm in, we tend to think about convergent evolution, right? So you get sort of the same morphology, in really disparate groups because they've come up with the same sort of solution to adaptation via like a lot of different mechanisms. So I think that's one particular way to address the issue is that, evolution can tinker with different parts of the genome and end up with a pretty similar phenotype, right? And there's a lot of interest also in thinking about parallel evolution and the predictability of evolutionary change in that same way. And that is like multiple solutions to the same environmental or ecological problem. And the answer is yes, we see them all over the place, which is really exciting and also complex.
From implementing the quantitative analysis point of view here, I think this kind of gets back to that idea of what is your comparison, right? So thinking about, well, are you looking at, a p53 mutation versus, whatever the population normal, wild type p53 allele is? Are you looking at a set of combinations of, different exposures versus a second set of combinations, or how are those things differing? Is really going to be important when you're trying to actually like estimate some quantities there in terms of how much does this cause that.
I would just add that I think the idea of having multiple causes for the same effect, the results, as far as I understand, and I think we already have the results for that in principle. Then there is nothing specific about having one causal effect versus multiple variables and the effect of this vector or this set of variables on the outcome. Then in principle, in theory, this is already possible. I would say the two challenges, the bottleneck is as far as I can tell, is in the modeling. Whenever you have a complex system, the modeling that is indispensable and there's no way of escaping becomes much harder. Then I would say we didn't even reach in some way, we didn't even reach the state of estimation, even though it would work, because the modeling, at least from the subject specific, the people that understand the field, they are not, as far as I can say, but it depends on the field, they are not doing that yet. Anyhow, this is one problem. I think the bottleneck is in this one step about modeling, but the theory works for sets in the same way. The second bottleneck that I think is interesting, that is a topic that I've been thinking about in my lab for a few years now, and we don't have a paper yet, but is the idea about when you have multiple causes, how can you have some type of summary of these multiple causes? Because you can kind of try all combinations of the causes and the effect of X1, X2, and X3, on the variable y, x1 and x2 in the variable y, x2 and x3, and you can do all the combinations, and you have this bunch of distributions that we can compute, but it becomes very hard to interpret what is the real effect. Then I think this is an open research question, I would say. There is some kind of tip of the iceberg on something called Shapley values that people are trying in the literature of AI, and they call explainability or interpretability, but I wouldn't say that I cannot claim that, at least I haven't read a paper, and I don't think that we have the solution yet for the causal portion of this this problem in the multivariable or high dimensional setting. I think it's a beautiful problem. I hope that it makes sense.
Thank you. Thank you. You got a request to write that paper. All right, I'll take a question from Slido, and that is, at what point would our tools and methodologies be able to reverse engineer causality? For example, using deaths of unknown etiology to extract public health insights. Anybody want to take that one?
I mean, in some ways, we've been doing that since 1600s. We're always just using the data to try to figure out something that goes on. And, starting with John Grant's on the bills of mortality in 1665, or 1665, where he tried to look at, what is the frequency of deaths, and then does this support the hypothesis that certain infectious diseases are more common in the first years of the reigns of kings and things like that. In some ways, this is all we ever do.
That sounds like a fascinating discussion to have further and deeper on, Ellie. Thank you. Nathan, Nathan, go ahead.
Yeah, thank you. Just want to ask another question. So I'm thinking primarily of some of the things that Elias brought up, and maybe others want to get into this as well. First, I just want to say I'm a huge fan of the work that Judea Pearl and the group that you were a part of all put together. The Book of Why, I think, was just brilliant. I basically made all my students and postdocs read it, or at least I pushed them pretty hard when it came out. We started a discussion group around it and all kinds of stuff. I think it's really important because it is this push towards not what's easy to compute, but what are we actually trying to answer? And I was quite convinced by the arguments in there that that's an incredibly important avenue that obviously all the speakers are getting at through causal inference, but it's really important. So my question is, I think that the difficulty has always been the degree to which you have to have a model, right, or really build something up so that you can actually ask a counterfactual and get at that third layer, which is so hard to get at. And the thing that has struck me a lot recently, and I'm just sort of curious if you would agree with this, or if the panel would see it the same way, is I think some of these advances in, for example, digital twins, which I alluded to a little bit before. One of the things I love about those, and we have built one of these for brain health, is that when we get into this kind of space, you can actually simulate the clinical trial in advance, and you can simulate outcomes under all kinds of different settings, if they'd been younger, if they were older, if you had chosen a different intervention pathway, et cetera, through a sort of causal model. And I'm just sort of curious if you see the development of that field as being sort of the piece that's needed in order to push forward a lot of the causal inference under kind of more that Judea Pearl model, or not, or if I'm not seeing that correctly, or if you agree with that, or I just love some conversation around those kind of issues, because it strikes me as that we're entering a different period in terms of what might be possible from the, getting at causality in some of these other ways.
Yes, thanks for the question and the kind words. The picture, the figure that I showed, and I will not do the whole thing, but I don't know if you remember this figure, if I'm allowed to share. But this was the figure that I shared at some point during the talk, and the model in the right side can be considered the twin, if you wish, and now the question is about, and it applies exactly like to the digital twins. I will talk broadly. I'm not familiar with your particular version, but I would say there is this literature about trying to do the twin. And the twin, at least the non-causal twin or the standard twins, that I have seen, they usually are trying to mimic whatever data you have. Let's say if you have observational data from the left that is red, I will call a twin if I'm able to make the object in the right, the observational data from the object in the right that is M hat to match the M star in the left. And then at least people, the standard ML, people that haven't heard about causality and in different fields as well that are start talking about twins, they'll say victory. And it is hard to do that to work, by the way. I teach ML, AI, and so on, maybe taking 1 month to make these things to match. I use a high dimensional model. It's a nightmare to make these things to match. Nightmare means very challenging. It's very finicky, the kind of model. But now, in some way, the problem, it is still the case that the assumptions that you need are not necessarily here in the right. The only thing that we did is to match the observational distribution. Then the question that I was trying to answer in this slide is about, what about if I do the inference about the effect of interventions in the model in the right, or about the counterfactuals that is layer 3 in the model in the right? What would we expect? And the answer is like, we cannot expect in general to get the answer as if it's the guy in the left. Because we still, and we can prove that mathematically, by the way, we'll do that, I can share papers and so, but we still need the same kind of causal assumptions, or how people call it machine learning, inductive bias in the neural net in the right, in order to solve the problem, to see if it's identifiable, not identifiable, boundable, and so on. Then I'm totally in favor of the literature of digital twins, or having these neural models that are proxies or surrogates to the model in the left. But the core issue still remains. That is, we still need to do causal modeling, elicit the assumptions, and then find a way of embedding that in the right side. I don't know if I hope that this makes sense. But this was exactly the point.
Yeah, I'll just add one little piece there, which is, it makes total sense. I think it's really important. I should maybe be a little more explicit when digital twins. I'm thinking more of like systems of biology type models where, we've maybe taken mechanism from 1,000 papers in the literature, right? So you can actually do forward calculate. You're not just doing statistical fitting in a sense, right? You're actually have a mechanism where you can then say, if we tweak the binding parameter of X, like what does it do? Right. And then how many fewer patients would we expect to have this disease if we hit this target at this level, et cetera, that kind of thing. So I think there is an element of mechanism.
If the mechanism are the quote unquote true mechanism or a good proxy for the tool, yeah, I think it's awesome. That's perfect.
Yeah, so I want to jump in here too, because I think this is another place where thinking about this idea of like, what is the ideal way that we would study this question? And then how are we emulating that with either a trial or an observational study or a simulation study can really help us think about what we need to assume and how we need to frame this. Because any kind of simulation study and things like studies using surrogate controls and digital twins have a simulation component, and so kind of fall into that. All we have to do to get a comparison of counterfactuals is to change some input in the simulation. But we don't want just any counterfactual comparison. We want comparison between the counterfactual under some treatment and what would have happened in the real world, right? We need those counterfactuals to be tied to the real world. And so thinking about, is our simulation model able to replicate causal effects that we see in the real world when we estimate those from data? Also thinking about, are we properly capturing all of the assumptions? And basically the idea is like, when you do use a digital twin or some other simulation approach to try to emulate that trial, you're much further away from the hypothetical ideal of the trial. And that distance means that you have to make more assumptions. And this includes things like the structure of the simulation model. It also includes things like the quality of the data that you're getting the inputs from. So when you take those sort of million other samples and get an estimate of a parameter value from them, has that estimate been done in a way such that it itself is a causal effect? And is that causal effect transportable or generalizable from that setting where the data was collected to the setting that you want to ask your digital twin questions about? And Elias is right that if we don't have the right set of assumptions, we can't expect to get the right answer. And in our group, we've done a lot of things looking at, okay, what assumptions, if we do have the right assumptions, do we get the right answer? And that's correct. And then also, what additional assumptions do we need to think about emulating these trials with simulation models rather than observational models? And a lot of those are around much more complex generalizability, transportability type assumptions, which are a sort of a cutting edge, forefront of the field in terms of methods that are being developed now in causal inference. So I think that there's definitely the possibility to do it, but there's a lot of sort of groundwork that needs to be done as well in the methods side in order to come up with some best principles on some of these things and understand exactly what we're doing and how we need to do it.
Thank you. All right, I see Lucila's hand is up now. I know that we probably have about five or 6 minutes left. So if we try and keep responses quickly, and Wenying and Ben, I'm going to come to you for some parting thoughts. So please be ready, Lucila, please.
I would like to use the brain power in this room there to answer a question that I often get. Is how do you see the value of synthetic data?
Wenying, please. Yeah, she's
okay. So I think they are extremely important, right? Because when you analyze real data, you do not know the ground truth. But when you have a specific model and you know the ground truth, you generate data, and then you can then from these data you can also test. What you can also ask, for example, what your sampling frequency, how much noise can your method tolerate, and if you make the structure say more complex or less complex or intact, species intact one way or another, how would it affect model performance? So I think they are extremely important.
Yeah, and let me clarify, because I completely agree with you on the synthetic data with the gold standard that you produce. But people are often in the medical field talking about synthesizing patients, trying to emulate patients so that you can do some models with them without having to do a whole lot of IRB approvals and deal with real patient data. And I won't tell my answer, but I'm hoping yours will confirm what I keep telling this.
This is also related to what I was saying about simulation models, because basically you're simulating fake patients based on what you think you know. And so that simulation is only going to be as good as the quality of the information that you think you know. I think it can be useful if you really have some good sets of assumptions that you really believe on, and you have some good core knowledge to build those synthetic patients from. But it's always going to require more assumptions than actually having the data. And there's a risk that you accidentally conclude that something is an output when actually it was an input, like you built into the model that it would always tell you something, but now you're looking at the model results and saying, ooh, I concluded this. And that's always a problem with simulations and synthetic controls.
Yeah, so my answer was always, if you have the real data, just do the extra work and use that. But thank you so much.
Thank you. All right. I'm going to end on a final question. So I want you all to think about this before I am going to turn over to Ben and to Wenying for a couple of parting thoughts. But my final question is going to be from Slido, which is there any reference book to guide me to include some of the topics discussed into my quantitative courses, undergraduate level, or to create a new course? This is from a person who teaches biostatistics and computational methods for biologists. So I want you all to think about that and have a quick answer. Now I'm going to turn to Ben. You've heard this conversation. You've picked up on some of the big points. I want to give you an opportunity to respond to anything that you've heard.
I'll respond to that point and then make a more general comment. So first of all, a number of people have already mentioned the Book of Why. It's a great book. So I recommend that. I actually have an open source research methods course for undergrads that has a lot of materials about causality as well. So you could check that out if you're interested. In terms of sort of closing thoughts, I mean, I think, causality is so complex and so broad. And as you saw from the panelists, there's people are approaching this in so many different ways. And there's, cutting edge techniques that I think are important to be aware of. And at the same time, I think we have learned a lot just in the past 30 years or so And again, going back to the Book of Why, some of the core things that I think we've learned about, like, why do we need to control for certain types of variables and why do we not want to control for other variables? We have a pretty good sense of that, actually. But I don't think that that knowledge is like widely understood. Like, statisticians don't teach it from a causal perspective, but these questions are really fundamentally causal. So I think there's a lot of work to be done from the educational side, even about sort of like basic concepts in causality and how important they are for all data science, basically.
Great. Thank you, Ben. Wenying, you've heard the conversation. I want to give you a shot as well, and then whatever your textbook or resource or paper is.
So I feel that for, like I was trained as a biologist, so I still feel that for us to understand the field, we have to have math foundation. And I would even say, right, like the undergraduate level, even if you was a math major, it's still not enough, right? Because I think the causal inference field needs more, because it's from such a diverse, so many different sources. So I feel like I still feel that somebody who is able to translate those highly technical literature in a way that's accessible to people with, like, say, bachelor's degree in math will understand. I think it's very critical.
Thank you. And is there I'm sorry, I missed was there a resource in particular that you were suggesting? As a tool to answer the question from the Slido, which is there any reference or book to guide me to include some of the topics discussed into my quantitative courses?
Yes, I mean, so depending on the background of that person, right? Depending on what is your background. If your background is still in say in biology, I would still argue that you would need to know the basics of the basics, right? Which is, start with calculus, linear algebra. That's just not avoidable. Calculus, linear algebra, statistics, and probability. That's not avoidable. And then the more advanced one that really takes, like what Alex did, right? Like, not being afraid when you get into new fields, like, say, topology. I mean, not be afraid of reading those books on, or even Julia Pearl's books on, causal inference. I mean, not be afraid of that. And so that's what I'm saying. Like, there need to be some source, which I'm not aware of, right? Some source that can ask, I don't know, like serving as TAs, right? So if you have questions, you get stuck reading a book. Whom would you reach out to, right, to address these questions? So I'm not sure. So that I'm not giving you a great answer. I'm wish list.
That's all right. The book needs to be written, it sounds like. All right, Jose, Rebecca, Ellie, and Elias, I'm going to ask you the same question if we do it relatively quickly. Jose.
Yes, so I would say that a wonderful, less mathematical yet rigorous introduction to causal inference is the book Observation and Experiment by Paul Rosenbaum.
Terrific. Thank you. Rebecca.
Rebecca's
got it in her hands.
I like Bill Shipley's book, Cause and Correlation in Biology. Found it to be very readable. There's a good bit of math, but it's beautifully explained.
Perfect. Thank you. Elias, one reference.
I already shared I'm writing a book as well. Hopefully I can share it soon. For many years in reality, but I'm sharing the PNAS paper, the PNAS paper that we have in data fusion. Maybe there are many links there. Then there's a little survey that could be useful. Thanks.
Terrific. Thank you. Thank you. That's great. And we'll shameless plug for the book when it comes out, please. And then Ellie, final word. What would that reference be?
Yeah, I would encourage people also to check out Causal Inference What If by Miguel Hernan and Jamie Robbins. That book is sort of aimed at people with less of the mathematical background, although there certainly is math, and it has also sort of more practical, how do you actually do things? I think the Book of Why is a great resource for sort of how should we think about these things, but then Causal Inference What If helps with, like, okay, now what actually am I going to do? So I would recommend that one.
Terrific. Thank you. Thank you for living up to incredible expectations about a really interesting, fascinating conversation. One that's really important and becoming more important in biology and the life sciences. And I'm going to turn it back over to our chair, Ann Arvin, having gone over a few minutes. So sorry about that. Thank you all to all the panelists.
I think we could spend another day and a half together, or maybe week and a half or more. But this has been a fantastic introduction for all of us. I think we will talk to Kavita and team about what we can do maybe to organize ways of addressing some of these very complex issues and communicate better about them.
So thank you all for joining us today. We're going to take well, we're done with our public session. And so for committee members, we'll take a 10-minute break and join back here at 3. Thank you.